The overall goal of this method is to identify the phenotypic properties and engraftment potential of clonal hematopoietic stem cell, or HSC, precursors during murine embryonic development.
This method can help answer key questions in the field of developmental hematopoiesis about the unique lineage contributions of clonal HSC precursors during embryonic development.
The main advantage of this technique is that hemogenic precursors are analyzed at the single-cell level. This allows identification of the unique phenotypic properties that distinguish rare HSC precursors.
Begin by adding aorta-gonad-mesonephros (AGM) tissues dissected from embryos between 9.5 and 11.5 days postcoitum to a 15 milliliter conical tube containing 10 milliliters of PBS supplemented with 10% FBS on ice. When the tissues have settled to the bottom of the tube, aspirate the supernatant and immediately add 1 milliliter of 0.25% collagenase to the samples. After 25 minutes in a 37 degrees Celsius water bath, stop the reaction with 1 milliliter of PBS plus FBS and use a 1 milliliter pipette tip to triturate the tissues 20 to 30 times until a single-cell suspension is obtained. Then dilute the cells with 8 more milliliters of PBS plus FBS and collect the cells by centrifugation. Resuspend the pellet in 500 microliters.